Good afternoon all. So today's topic is about Pili and Fimbria. These two topics are coming under the bacterial cell structure. So what do you know about Pili? The Pili, it is a plural form of Pilus or Pilus. You can say any. I usually say Pili or Pili. This pilus is a hair-like appendage which are found on the surface of many bacteria as well as archaea. It is another group of microorganisms. The term pilus and fimbriae, this have been originated from the Latin word for fringe. The fimbria word originated from the fringe and its plural is called fimbriae. So, it can be used interchangeably, although some researchers reserve the term pilus for the appendage required for the bacterial conjugation. So, both of them are similar in the character, but the size is different. I think Fimbria is smaller compared to pilus or pilus. And this pilus is used as an appendage for bacterial conjugation, whereas Fimbria, it is used for the adhesion. That means attachment of the bacteria within the surfaces. All the pili in the later sense are primarily composed of apelin proteins. So this pili, it is made up of apelin proteins which are oligomeric. Some bacteria, viruses or bacteriophages attach to the receptors on the pili at the start of their reproductive cycles. So there are certain bacteria, viruses or bacteriophages, which means the virus which attack the bacteria. They attaches to the receptors which are present on the surface of the pili on their reproductive cycles. And the pili are antigenic. That means it is capable of producing antibodies. Antigenic. This term antigenic etc. will learn in the later, that is in the immunology. Okay. And these pili are also fragile and also constantly replaced. It is very fragile structure, so it can be constantly replaced, sometimes with the other pili of different composition. And so as a result, what happens? It results in altered antigenicity, which means the original pili, it is different from the replaced pili. Both of them are different in their antigenic composition. And also it may result in a specific host response. So what is the character of this antigen? Antigen in the body is capable of certain host responses. That is, the production of antibodies or any other type of host response will occur as a result of this antigen binding. So in this case, specific host response to old pili structures are not effective on the new structure. So old pili which are capable of inducing certain host response which will be quite different in the new pili. So that response is not same as to the new pili structure. Okay. Then the pili which may consist of certain type of genes which are called recombination genes. That means recombination genes of pili, it codes for certain variable and constant region of pili. Both this variable and constant regions, these are present especially in the case of this antibodies. Antibodies, if you are studying the structure of this antibodies in immunology, you can study that this antibody possesses both variable as well as constant region. Actually, this antibody is what? Protein. So, this antibody possesses variable and constant region. Like that, the recombinant genes of this pili, which codes for the variable and constant regions of pili. These terms which you will later study in the immunology. Now, Pili acts as primary antigenic determinants, virulence factors, and on the cell surface of a number of gram-negative and gram-positive bacteria. So, in certain gram-negative as well as in the gram-positive bacteria, this pili acts as a primary antigenic determinants. Pili acts as a antigenic determinants, especially the bacteria including in the family Enterobacteriaceae, Pseudomonasaceae and Neisseria. Last CEAE, which may denotes the family. Okay, this CEAE denotes the family. Okay. Next we can discuss about types of pili. First one is conjugative pili. So what is the peculiar character of this conjugative pili? Those conjugative pili allows the transfer of DNA between the bacteria. So when two bacteria are present, if the one bacteria wants to transfer its DNA to the other bacteria, and that process takes place with the help of this pili, and those process is called bacterial conjugation. So in the process of bacterial conjugation, for the transfer of DNA from donor to the recipient bacteria, those takes place with the help of a pili. So they are sometimes called sex pili in analogy to sexual reproduction. Why? Because they allow the exchange of genes from the donor to the recipient. Through how? Through the formation of mating pairs. Perhaps the most well studied is the Pilus of Escherichia coli or E. coli. So this organism is mostly studied. This conjugation process is mostly studied on this organism. The pili is typically, what is its length? 6 to 7 nanometer in diameter. So it is very much small, only observable through electron microscopy. In the case of the light microscopy or no other, we can't see this structure. So what happens during conjugation? During conjugation process, a pilus, a passage, a pilus which is emerging from the donor bacteria, it moves towards the recipient bacterium and it comes in close. That means draws its close, and this eventually triggers the formation of a mating bridge between the donor bacteria and the recipient bacteria, which establishes a direct contact and the formation of a controlled pore which allows the transfer of DNA from the donor to the recipient. This is the process of conjugation, and this conjugation takes place with the help of this sex pili. Typically, the DNA transferred from, that is the donor DNA, it consists of certain genes, and these genes may be sometimes encoded on a plasmid. Plasmid, which means what? Extra chromosomal genetic material. However, there are sometimes other pieces of the DNA, often co-transfer. The co-transfer of other pieces of DNA may also occur. So what happens if the other pieces of the DNA are also co-transferred? This may result in dissemination of genetical traits throughout a bacterial population, and this may be a reason for antibiotic resistance. That means this peculiar character of this antibiotic resistance, which can be transferred from the donor to the recipient through the conjugation process. Not all the bacteria can make conjugative pili. So, every bacteria are not capable of making conjugative pili. There are only some bacteria are capable of making conjugative pili. But the conjugation can occur between the bacteria of different species, not only among the same species, but the conjugation can also occur between the different species. Next, another type of pili, that means type for pili. Some pili, which is also called type for pili, the short form is T for P. It generates a motile force. What is the motile force here we are mentioning? The external end of the pili, that means a pili which is emerging from a bacteria which adhere to a, means attaches to a solid substrate, either the surface to which the bacterium is attached, either to the surface where the bacterium is attached, or this pili can be attached, this external end of the pili can be attached to other bacteria. Then when the pili contract, and after this attachment the pili contract, and this may result in the pulling of the bacteria forward, and it resembles like a grappling hook. This is a type of movement. So this may result in generating motile force. The movement produced by this type for pili as a result of this contraction of this pili on the contact with certain surface, it is typically a jerky movement. So this type of jerky movement is called twitching motility, as opposed to other forms of bacterial motility. So it is this twitching motility, it is quite different from other type of motility which are produced by the flagella. And also there are some bacteria, for example Myxococcus xanthus, they exhibit gliding motility. It is also quite different from the twitching motility. Then bacterial type for pili are similar in structure to the component flagellines of Archaea. It is a type of flagella which are present in a archaebacteria. So they show resemblance with this bacterial type for pili. Next is about Fimbria. So this Fimbria and pili are similar, but some difference are there. They are also called common pili or adhesins. Actually this Fimbria is a short pilus. Pilus, which is a long, which take part in bacterial conjugation, but in the case of this attachment or in the adhesion, the pili is called now Fimbria, which is a short, that is used to attach the bacterium to a surface, that means in the adhesion. So they are sometimes called attachment pili. So this type of pili, that means Fimbria, are also called attachment pili or common pili. Fimbria are either located at the poles of a cell. So within the cell, they may be located at either at the pole of the cell, or they may be evenly spread over the entire surface. So within the entire surface, sometimes these Fimbria are spread, or sometimes it may be present at the pole of the cell. But these are not visible in the light microscope, only clearly visible under the electron microscope. And there are certain mutant bacteria that lack the Fimbria. Those mutant bacteria who lacks those Fimbria, they cannot adhere to the usual target surface. So there are certain, so this function of this Fimbria is attachment. If those bacteria which lack Fimbria as a result of the mutation, they cannot adhere to the usual target surface. So thus it cannot cause disease. Such type of bacteria are not capable of causing disease. For example, Neisseria gonorrhoeae, the organism which is gram-negative Coccobacilli, which is responsible for causing gonorrhea. But this bacteria, as a result of this mutation, they might lost their Fimbria and now they become non-pathogenic also.